Well, I think the trouble is that people regard science as something over there rather than something that affects their everyday life. And that's true of people actually working within scientific industries as much as it is of the general public. So I think it's really important that we try to not talk down to people about science, but talk with people about science, but understand that their background may be that they don't really have a detailed knowledge of the science behind it. So we have to fill that in a way that's efficient. So I like to take a subject like, for example, stem cells, or for example, epilepsy, or maybe nowadays coronavirus, which is I'm spending a lot of my time giving presentations to different groups, to Rotary societies, to all sorts of people, undergraduates, graduates, and actually, I think pictorially is very important, though. Just listening to somebody talking is one thing, and that sort of Welsh preacher stuff can work at times. But frankly, I think having some illustrations that people can follow without actually listening too much to the words is really, really vital. So what I tend to do is not necessarily give them a sort of hose pipe flood of stuff, but put lots of pictures up. So instantly people are understanding what a virus may look like, how it gets into a cell and so on. So I think that's really critical. People get rather cynical or afraid of some science, particularly in my field of life science and medicines. And you can see quite recently with the COVID vaccine, the sort of people who are very concerned about whether it's safe and so on. Indeed, then you get all the wild stuff about, is it a way of spreading political diseases and so on. So I think it's really critical that we try to explain things in a way that doesn't mean that we have all the answers. Science doesn't have all the answers. In fact, what always has motivated me is a Swedish philosopher's quote, Paul Anderson. He said, "I've yet to see a problem, no matter how difficult, when you look at it the right way, it doesn't become even more difficult." And that's kept me going all my research life. So I think that's the first thing, to demystify, if you can, some of the problems, so that we can actually talk about the opportunities that it presents to the patient or to the public at large. The other thing is that for a company, it's really important that people everywhere understand what they do. I mean, it's Henry Ford, wasn't it, who used to go down and get people on the shop floor saying, "What would you like to do?" And the chap said, "I'd like to actually see what happens at the other end of the line." He's doing his little job there. So I've found all of my life in my industrial career to try to talk to different sectors of the population. And you know what, sometimes, like when I was at the Boots company and I was developing ibuprofen, a very widely used drug now, to actually persuade people to write down the formula, the chemical formula. And I know I was in a pub one day when I saw this chap and he was writing it down. He said, "This is what I'm working on, you see." Now he was, he was, I think, just a very general cleaner. But the fact is, he felt a pride in what he was doing. And actually that's good for the reputation of the company too, because it spreads knowledge of that. So I feel that's really important, that all the workforce get involved in understanding what the business is, and not just the scientists saying, "Well, trust me because I'm doing it." We felt when we understood certain diseases centuries ago, we described them by a name, and then more recently, in the last 100 years, we've given names to things like cancer, as if it's a single condition. And, I tell my students, well, you all know what a cough is, but is that lung cancer? Is it bronchitis? There's all sorts of reasons why that symptom comes there. So it's really important, if you take something like cancer, to actually try to delineate it. Is it a blood cancer, solid tissue cancer? And even then, why do those cells suddenly go crazy? Why do they take off and reproduce themselves? The quality control mechanisms got lost in there somewhere. Yeah. So we then start picking that apart and looking at all the pathways in there. And drugs have classically been discovered by saying, "Oh, there's a target, there's a pathway there, I just interrupt that and I can kill it." When I developed AZT, my team, the first ever drug for HIV, we knew that the virus had to reproduce inside a cell, and it had to take some of the ingredients of the cells to do that. And we put a chemical that looked like one of those ingredients, but was different. So it just couldn't cope with reproducing it, and it stopped it. So that was the way we did it. We thought single target, single drug, and it works. Well, it doesn't work that way. There are many different pathways, as we were looking at with cancer. So what's happening nowadays, a company that I've just become chairman of, eTherapeutics, for example, we look at all the networks that are taking place in a cell in the brain, cell in the skin, cell in the kidney, and then we can actually ask the computer to look at how these interrelate. Then I can actually ask the computer, if I put this chemical or that chemical or the other chemical, does it sort of blow apart that network? Not that it hits a little track in it, but it blows it apart. And now I'm onto something quite interesting. But that's a much more complex pattern of understanding. I'll give you another example. My team developed a drug for epilepsy. And when you have epilepsy, one of the symptoms, one of the things is fits, of course. And this is because a chemical in your body called glutamate suddenly floods, and you get this, movement of the limbs. Well, we thought if we could block that glutamate, then it could stop that. And indeed, this particular medicine called lamotrigine does that. So we launched that, we did all the studies in people, showed it reduced or sometimes eliminated all these fits and seizures. And I was going around the world showing how these neurons fire and how we block that glutamate. Postgraduate student came to see me a year later and said, actually, what it does, it works somewhere in the brain on blocking sodium channels. So, well, no, that's not. He said, it does, because that is the precursor. That's the starting point, which allows these glutamates to flood. And yes, of course, you're blocking it. But if you can only go to the source, the tap at the top there, then actually we probably have an even better drug. So it's that understanding of complexity, and that it's not a mystery. It's that we have to really start to learn more and more about that. I think it's reasonable to try to pretend that these things are simple, especially in life sciences and the processes in the body that go wrong and the ones that go right. I think it's important, though, not to try to impress people that the subject by using long hyphenated words. In fact, I just got one here fortuitously. I didn't realize I was going to use this today. This is the opening thing in a letter. Whilst I was a postdoc in 1964, I designed 2-cage ligands that would require a central metal atom to adapt respectively the less common 6-coordinate triagonal prismatic and 8-coordinate cubic configuration. What? It's important that not to say we don't know, and we don't know much about all the disease things, but what do we know? I think we should try and talk about what really happens, in a cancer cell, in epilepsy, in a coronavirus, and then get people to understand that, well, it's a step along the way. You see, look, we only cure diseases, infectious diseases from bacteria. It's the only thing we cure at the moment. All the rest we treat. Now, what's been happening in the last probably 5 to 10 years, as we started to understand which are the genetic linkages here, what are the genes in our body? It's not one, the multiplicity of these that are involved. Could we actually pull them out and stick the right gene in, the one that's gone wrong? And this is an amazing new science called CAR-T, C-A-R-T therapy. So you probe the body and find where the DNA and so on, you find where along that strand, there's something that isn't normal and is related to this condition. You then go into the laboratory and you make a vector, a virus or whatever it may be, put the right gene in, and it seeks this, clips the old one out, puts the new one in. Now that is going to cure a lot of diseases. What we're doing with the coronavirus, of course, is cheating the body. What we've done is little spikes on the surface, the corona, the crown. Take those spikes away. They can't make you ill, but they can tell the body that there's something wrong there and it's going to attack it. So we wrap those up in another harmless virus or a little emulsion, put that in the body thinks, what's that? I don't like it. And it grabs hold of it and the antibodies arrest it and the T cells kill it. So, it's that science now that we're understanding at the genetic level, the gene level. But if I do that, I've got to be pretty sure that's the only thing I'm doing, correcting that one gene. Otherwise, I'm going to do friendly fire all over the place and another nose appears here or whatever. So this is not without its difficulty, but that's a very long way from, oh, this plant in my garden, when I eat it, makes my heart speed up. Oh, digoxin. It's a long way from that kind of chance discovery that a plant has some medicinal properties, to actually understanding the mechanism in the virus in the cancer cell, and then correcting it by, if you will, genetic surgery. Certainly this whole area of gene manipulation, gene therapy, gene replacement is, to my mind, where we're going to see the biggest advances. But that linked to artificial intelligence, linked to, really trying to map these things in a way that we haven't been able to do in the past. The computers are there, and we've used them successfully to sift out molecules and so on. But this, combining our understanding of genetics with artificial intelligence, can make a big difference. I've always felt with students, well, not students of all age, but particularly those starting their career, that there comes a point beyond where they've got to focus their attention on a narrow subject to get their examinations, to actually let knowledge drift over you, listen to different kinds of science in different places, not just the one that's your speciality, and actually suddenly grab pieces of that. And then you'll find that really, really becomes a very exciting thing, because you're trained to think scientifically, but you're looking at other areas. So it's really, I suppose the answer there is very simply, don't just focus on what you think you know, get a broader view of life. And that's really important, I think, in choosing their career. We don't know what we're going to do 10, 15, 20 years, and we surprise ourselves at times what we do. But I think thinking ahead for a few years, enjoying it, and then moving on is really, really, really critical.